Thank you, Dr. McCullough, for that very generous introduction. I'm sorry I can't be with everyone in person. The weather had other plans, as I think many of us have learned. So what I'm going to do today is try and balance the sort of usual approach to a stroke genetics talk, where you go through lists of syndromes and how one might think about identifying them in the clinic, with the sort of forward thinking about what it is that we who take care of patients might anticipate in the coming years, how genetics might become useful. So my outline is just to briefly introduce the audience to how we think about rare variants in the genome versus common variants. The rare variants are those that cause Mendelian forms of stroke. And then I'm going to spend most of the time on the common variants, which are what we've begun to identify through genome-wide association studies. And I think this will be my way of introducing you to what I hope will become the future. So here you have a glossary of terms. It should be in your handouts, just to keep us straight. Nothing probably that you aren't familiar with already. So the first concept I think that's fundamental to understanding how we in genetics approach understanding the role of genetic variation in something like stroke is to recognize that gene variants or genomic variants basically have two fundamental properties. One is they can either be common in the population or rare. And you see this along the x-axis here. As frequency goes up, you move to the right. And if they have an effect on a trait by, for example, increasing your risk of stroke or decreasing your risk of stroke, that effect size can be greater or lesser. And in general, the common variants have relatively small effects on a trait, while the rare variants that we identify as causing inherited, autosomally dominant or recessive traits, stroke syndromes, they will obviously have a very strong effect size. And that makes them, once you've identified them in general, it's much easier then to move from rare variant discovery to mechanism, whereas once you've identified a common variant associated with a trait, it's quite challenging to figure what the mechanism is that explains that association. So let's just back. So when we think about rare genetic stroke syndromes, they're basically either stroke can be an associated feature, but in general, it's a final common pathway where you can have a disruption in any biological system within the body that leads to increased stroke risk. So examples can be rare cardiomyopathies that give rise to cardioembolic stroke, clotting disorders, collagen vascular diseases, blood dyscrasias, lipid disorders, kidney disorders, or bioenergetic disorders. And these disorders that are outside of the brain, if you will, and the brain vasculature, make up the majority of this list of Mendelian causes of stroke. And this list is only partially complete. But then there are loci, or the regions in the genome that harbor rare variants that give rise to stroke syndromes that are specific to the blood vessels of the brain, the COL4A1 or A2 inherited small vessel diseases, or the Notch3 disorders. And what we've been learning is that many of these have not only a rare Mendelian variant within that gene, but also harbor common variants that exist across the population that are associated with a very small increase in the risk of stroke. So I'll give you two examples of these. These are the Mendelian causes. One is CADASIL, which exists both, you can see, in children and in adults. It's caused by autosomal dominant mutations in the Notch3 gene. There are quite a few that have been identified, and the result is you get misfolding of the Notch3 protein, and you have consequent derangement of vascular smooth muscle. You'll identify these variants in about 5 out of 100,000 individuals of European ancestry, and patients can present with migraine, stroke, psychiatric symptoms, progressive dementia, but in general, there will be very strong and clear family history. Another example is Fabry's disease, more common in the population. Here you have alpha-galactosidase deficiency, which leads to accumulation of glycosphingolipids in multiple tissues. The prevalence can be as high as 0.5% of people who present with stroke under the age of 55. And in addition to stroke, you can see neuropathies, GI distress, corneal opacities, hearing loss. This is an X-linked disorder, and it presents earlier and more severe in men, but up to 70% of women with one abnormal copy of the gene can exhibit symptoms, although they are generally more muted than they are in men. Now we're going to focus the rest of the talk on common variants. And the motivation here is because we know that family history is a strong risk factor for stroke. Epidemiological studies consistently demonstrate that environmental risk factors, acquired risk factors cannot explain the burden of strokes. Rare monogenic syndromes obviously don't explain most of sporadic disease. And so the motivation is to identify these inherited factors that will help us understand biology, and then importantly, identify new targets for drug treatments. And we've seen some pretty impressive examples already in the world of lipid lowering and PCSK9 inhibitors. And it's important to remember that although statins were not discovered through human genetics had to catch up. But had human genetics come first, statins very likely would have been discovered through the association of common variants with hyperlipidemia in that pathway. So the very first common variant approaches are studies that I think we're all familiar with. We call these candidate gene approaches. And this is where you pick a region of the genome and you say you think that the gene there or the family of genes within that region harbor variants that influence risk of stroke. This has basically been successfully exploited in rare disorders, in which case these are situations where, once again, you have very high potency variants that cause familial syndromes. But in sporadic stroke, where there have been a number, and I've just listed a few, candidate genes investigated, they've been, I would say, a complete failure. And a failure for a couple of reasons that haven't changed over the last 10 years. The most important is, as you can see from the maps on the lower part of this slide, that the frequency of these common variants varies across the population. And what you see here is a map of Europe, and the blue circles represent the allele frequency for the ApoE4 variant in the population. So you can see that as you move south, in general, the allele frequency of E4 in the population drops, and as you move north and east, it rises substantially. So what this raises is the challenge that unless you know specifically the backline population of your cases and controls, you don't know whether you simply have an overrepresentation of individuals with ancestry, let's say, from all the way in the Northeast among your cases, and that explains the higher allele frequency of E4 that you find in cases versus controls. And until genome-wide assessment became possible, you couldn't really control for ancestry. And so there were tons and tons of false positive results, and probably equally numbers of false negatives. But the candidate gene studies basically were never replicated. Just trying to get, okay. So what is a genome-wide association study? If you look at if you think about how a common variant arose in evolution, it makes it easier to understand. So on the left here, we have, let's say, 40, 50,000 years ago, when the SNP, the blue SNP, was all that existed. And then at some point over the course of evolutionary time, there was a mutation that yielded the brown SNP. And because the brown SNP didn't have much of an effect on selection, or perhaps it had a slightly increased effect, improved natural selection, it became more and more frequent in the population. But let's assume that it really didn't have any effect on selection. This is just randomly, as the population expands, you can see more and more people have the brown variant. Then once you get to being able to perform a GWAS, you look at the number of people who harbor the brown variant versus the normal number of people who don't, and you simply do a count. And if you find more of those brown variants in cases than controls, you make an assumption that the brown variant is associated with the phenotype that you're studying. Let's assume being a stroke case as opposed to a control. When you do this, let's say for 1 million SNPs, you yield what's called a Manhattan plot. And what you have there on the x-axis, you can't quite read it, but each chromosome is represented. And then on the y-axis is essentially the higher the dot, the lower the p-value, and you get these stacks. And the stacks represent regions of the genome, so there are multiple SNPs in areas that are more frequent in cases than in controls. And then you set a threshold, which I'll come to in a second. Here it's that dotted line, and anything that rises above that threshold is considered to be statistically significant, accounting for multiple comparisons. So the challenges and opportunities for GWAS are that it's powerful but power hungry. So the guiding hypothesis for genome-wide association studies is simply that within the genome, there will be variants that are associated with the trait. So it's not that hypothesis. Some people in the field will say that means there's no hypothesis, but the key is that it's not affected by any preconceptions about biology or mechanisms. You're not suspecting one gene or another. Importantly, now we can control for confounding by ancestry because we can take advantage of the frequency, the allele frequencies across the whole genome to assign individuals an ancestry, and that allows us to control for that those differences I showed you earlier, where you have higher frequency of an E4 allele, for example, if somebody is from Northern Europe as opposed to Southern Europe. So now we can get around the failures of candidate gene approaches. But the challenge is that now you really have to be careful about assigning statistical significance, and so now you have the stringent control for multiples testing, and this has been established by convention as 5 times 10 to the minus 8, which is basically Bonferroni correction for all of the common variants across the genome. So now we're in the requirement of sample sizes in the 5 to 10 to 20 to 50 to 100,000 cases. And then the real conundrum has been, for those of us in this, that what GWAS has exposed is all of these non-coding regulatory variants that are associated with traits in ways that we have yet to sort out. So it's been very hard to go from all these associated variants, there are now tens of thousands, and actually link them to biology. But lots of work is being done, and there certainly is lots of progress to report. Sorry about the delay in the advancing. Okay, so I think it's important to take a step back and understand that the GWAS era has really been revolutionary beyond genetics. So this notion of introducing a real Bonferroni p-value can't be published anymore, essentially, without that p-value. Nobody takes an association seriously until you have achieved it, has led to incredible rigor and reproducibility in genetics. And it's really set the standard for human studies. I mean, who has, if you look at across the epidemiology literature, you look at other studies of human disease, it's very rare that we actually hold studies accountable for actually controlling for multiple hypothesis testing. But in genomics, it's the rule. The other piece is that now that we're measuring exposures that are SNPs, we can genotype SNPs with great reliability, whether we do it in Tokyo or in Africa or in New York City. A SNP is a SNP is a SNP. And there are very few human traits or exposures that can be measured with such precision and limited variability. The other thing about genetics is that we can generally assume that the SNP came first before the trait developed. So the direction of effect isn't one that we have to really confirm. And this also means that you can now test refined hypotheses once you have a genetic region identified because of a SNP in another phenotype. So once a SNP is associated with stroke, well, we know it has some biological impact on stroke. So testing its association with kidney disease means that we are testing a region of the genome with some impact on biology that is important to stroke, testing to see whether it also has an effect on kidney disease. So this allows you now to really define the genome as into biologically consequential regions. The other thing that the GWAS revolution has led to is really a transformation in the way we collaborate. So it's over the last 10 plus years, we've organized the investigative community around important questions and rigorous approaches, and organizing means hundreds of people, and actually now we're getting into the thousands. And the reason is, of course, you need to have large harmonized samples to achieve the sample sizes of cases and controls to study. It's led to, we've really had to rewrite some of the rules for collaboration and maintaining a scientific community. And of course, the result has been we now have biological targets for new therapeutics. We are now all focusing on trying to figure out which patients are most likely to benefit from treatments. And these consortia, I'll talk briefly about the stroke genetics consortia, have meant that there's really a lot of agreement over the priorities of the field. And that's there's much less infighting in the stroke genetics world now because of GWAS than there ever was before. And it's really been a pleasure to be a part of this community, but it also, I think, has led to our being much more creative. in exploring genetic hypotheses. So the red here represent countries, in some cases continents, represented by investigators within the ISGC, and this is always expanding. This was our first meeting in 2007. You can see a handful of us. This was at the AN annual meeting in 2007, and this was a recent one in Washington, D.C., attended by about 110 people. And our last in-person meeting was in St. Louis, attended by about 130 people. And we, look forward to getting back to our every 6-month workshops that are open to anybody who wants to come and participate. We've got a very robust junior investigator community, and there's a very welcoming atmosphere for anybody who wants to get involved, because at the end of the day, we need all the help we can get. So we have very simple convening principles that cerebrovascular disease is a complex disorder influenced by variation in many genetic and non-genetic exposures, each of which contributes only a small influence to disease risk. And therefore, large, that is larger than any single center can assemble on its own, well-characterized samples will be necessary to discover these exposures. And we are open to all who can contribute. We work very hard to recognize all contributions fairly in publications. So I've never seen an author list that was too long, in my opinion. And we work together in a spirit of cooperation, open communication, in order to promote the best science in the present and the best science in the future. And so these principles have generated an extraordinary amount of generosity, for example. So the investigators got together and have funded fellowships for investigators from developing countries, fellowships for junior investigators. This is really a volunteer-run society. We don't have dues. We simply run on contributions and registration costs for in-person meetings. We have a website, which I encourage all of you to visit, the Cerebrovascular Disease Knowledge Portal, where we share freely all the genetic data that IRBs permit us to share this way. We also keep the papers there, and so there are some summary level data sets available for folks to download and use, but the goal is really to share as broadly and freely as possible. And over the past 15-ish years, we've had, I think we're now in excess of 120 publications, multiple grant awards. Our workshop attendance continues to rise, but we've recently had to convert to virtual workshop attendance. And of course, all of you are encouraged to go to the ISGC website to see when the next virtual workshops are, if you'd like to join them. They're usually about 2.5 hours long and devoted to different topics of focus. So I'm now going to just give you a quick overview of the state-of-the-art ischemic stroke GWAS. It's about to be supplanted. There is so this is megastroke, and gigastroke is winding its way through reviews right now at a major journal, which will dramatically expand the number of SNPs or loci associated with stroke. But what you see here is that for megastroke, which was published in 2018, we assembled 67,000 stroke cases, 450,000 controls. To the right, it's just a flow diagram of the quality control process, and the different, and let's see, the fourth row down are the different studies that are representing different ethnicities, different ancestries. So you can see all the way on the left, EUR represents European ancestry individuals. Overwhelmingly, this was a study of individuals of European ancestry. Next, there are a few African ancestry studies, East Asian, South Asian, and so forth. So the first message is that this is really a study of European ancestry individuals, which is a very big limitation to these results, but we're fortunate in that more and more studies of mixed ancestry and different non-European ancestry populations are coming out. This is the Manhattan plot. You can see all of the stacks that exceed that dotted line are essentially representing the genome-wide significant hits. There are a few, the dotted line here is 10 to the minus 6 because that led to replication, but keep in mind that the standard dotted line is 10 to the minus 8, but there are some that have since been replicated. That's why the dotted line here is set to the 10 to the minus 6. So all of those stacks with labels are genetic loci associated with stroke. I think the main, rather than go through each one, some of them are very interesting. The main lesson here is that there was very striking overlap with other conditions. So loci associated with stroke, for example, there are 2 that are also associated with atrial fibrillation, 1 that was associated with carotid IMT, several that were associated with white matter hyperintensities, blood pressure, and so on. And then there are quite a few that are only so far associated with ischemic stroke. But this is reassuring because certainly you would expect ultimately that loci associated with risk of atrial fibrillation ought to be associated with risk of ischemic stroke. So the caution, as I alluded to, we have to continue to make sure that the megastroke loci that are identified in Europeans are also relevant to subjects of African ancestry. And conversely, we have to make sure that primary discovery is being conducted in individuals of African ancestry that can then yield loci that can be checked in European ancestry individuals. 2 articles, 2 recent studies listed here that have identified some of these limitations. The 2020 study from Dr. Keene and his colleagues identified a new locus in individuals of African descent that wasn't clearly identified in megastroke, and also didn't confirm a lot of the megastroke loci. So we really have to stay tuned. But fortunately, there are going to be a number of studies of individuals of African ancestry included in gigastroke. So I expect many of these questions will be addressed for the first time. So I'm now going to turn to intracerebral hemorrhage genetics, which has been my primary interest, really to tell a couple of stories and illustrate a couple of principles. The first is this, how studying all ancestries becomes important in understanding whether a risk locus is associated with a trait. In ICH genetics, it's a much smaller cohort than in ischemic stroke. These are various studies represented here at the bottom. They're all, with the exception of the European ISGC Europe, they're all from North America and mostly the US. 13,000 individuals. What we confirmed in this particular study when we asked the question about ApoE4 and its association with ICH, we confirmed that it was associated with risk of ICH in the lobar brain regions, which we'll come to in a second, but that's at the cortical subcortical juncture, typically associated with amyloid angiopathy. But then when we conducted a propensity score matching for hypertension burden, remember hypertension is a very strong risk factor for ICH generally, that E4 remained associated with lobar ICH in participants of Hispanic ethnicity, but was not associated with lobar ICH in Black participants, and it was not associated in any participants with non-lobar ICH. So clearly, we have more to learn. This was not a study that controlled for ancestry. It controlled for ethnicity. In other words, individuals were called Black or Hispanic based on their own self-report. But what you clearly see is that here is a gene locus that is very well established as a risk factor for lobar ICH, but clearly has a different role depending on self-reported race ethnicity. Now, given that, as we move now to applying genetic information into clinical decision making, we have to keep in mind that there are a lot of answers yet to be obtained in multi-ethnic samples. So these data are in largely white individuals that I'm about to show you. So as a bit of background, though, we think of cerebral small vessel disease as the most common cerebrovascular pathology. It's age associated, and it manifests in a number of ways, including, of course, hemorrhagic stroke, also commonly called lacunar stroke or small penetrator stroke, white matter hyperintensities on MRI, and then clinically, in addition to causing stroke syndromes, gait disorders, dementia, and depression. When we see a patient with ICH, we use location as a surrogate for identifying what we think is a subtype of the underlying small vessel disease. So if that hemorrhage is in the brainstem or the cerebellum or the deep territories such as the thalamus, we tend to think, or the basal ganglia as well, we tend to think of what many of us call hypertensive vasculopathy, or hypertensive arteriopathy, or arteriolosclerosis. And then if on the other hand, the hemorrhage is on the bottom right at the cortical subcortical juncture, the lobar brain regions, then we tend to think of amyloid angiopathy, although we certainly know that arteriolosclerosis can be associated with hemorrhage in those regions. These cerebral small vessel diseases are very important because they are progressive and they're common. And just to give you an example, here what we've done is we've summarized data on our ICH survivors. And this slide just is really set up to give you a sense of why cerebral small vessel disease is such a problem and such an opportunity. So what you have here on the x-axis is the years from first intracerebral hemorrhage. So t equals 0 is when the hemorrhage occurs. The red line represents Black and Hispanic survivors from our cohort. The y-axis represents deterioration in brain health measured as cognitive decline, dementia, or recurrent stroke. And you can see that for Black and Hispanic survivors, by 5 years, roughly half have developed substantial deterioration in their brain health. That figure is slightly lower for ICH survivors who are white. You can see it's maybe just under 40%. And then just for comparison, you can see that for individuals who have already developed white matter hyperintensity changes but have not had a hemorrhage, their risk is substantially lower, but it is not the same as the general US population modeled here in the green dotted line. So the way we approach the opportunity for ICH is we can, quote unquote, flatten the curves. That's our goal, because ICH for us is a warning sign. This is an opportunity really to intervene and to arrest cerebral small vessel disease progression. So there are lots of ways to think about it. We want to further risk stratify people. Now, it turns out that the number one thing we can do is blood pressure control, and we're really bad at that. So we have a whole program that really doesn't have anything to do with genetics that's devoted to figuring out how to improve blood pressure control for these ICH survivors. But blood pressure control alone isn't going to be enough. So how can we kind of combine decision make, all the clinical imaging and genetic data to make some decisions for ICH survivors? And this is a it's a much more focused question that we were able to ask. In this case, it was for survivors of oral anticoagulant-associated ICH. Those individuals are at particularly high risk because on the one hand, they're, as you saw before, at high risk for developing progressive cerebral small vessel disease, having another hemorrhage, and on the other, they in general have an indication for anticoagulation, most often atrial fibrillation. They're at risk for developing thromboembolic stroke. So we'd like to be able to really separate out those people who are at highest risk for ICH recurrence, because perhaps it's those individuals who shouldn't be anticoagulated and the remainder of individuals who should be put back on their anticoagulation. So we developed and validated a combined genetic imaging classification scheme, in this case ApoE genotype and MRI classification scheme, to predict ICH recurrence using card tree regression. And what you see here is we took 169 survivors of anticoagulation-related hemorrhage, and we began to analyze MRIs. In this case, CSS refers to cortical superficial siderosis, and then added on whether or not individuals had lobar microbleeds, and then threw in ApoE genotype. And what you can see here is we're able to, using these data, classify individuals into 3 groups: high risk, medium risk, and low risk. Those individuals who are low risk, starting on the right, were at particularly low risk for recurrent hemorrhage over 5 years, 0.6%. So they had no disseminated superficial siderosis on their imaging, fewer than 2 lobar cortical microbleeds, and their ApoE genotype was E3E3, which is the most common genotype. And you can see they had such a low risk of having another hemorrhage that they probably would be pretty straightforward as candidates for resuming anticoagulation. We then identified a group of medium risk, and those individuals, again, an intermediate risk of recurrent hemorrhage over 5 years, but probably they too, given a 2% risk, 2.7% risk, they might well be candidates for anticoagulation, 2.7% per year. And then the high risk group, which were identified by having the cortical superficial siderosis or more than 2 lobar microbleeds, regardless of ApoE genotype. That's the group probably they have a very high risk of annual ICH recurrence, probably would not be candidates for anticoagulation. But what you can see here is that using just the genetics, you're able to separate the low risk and the medium risk group, which could have pretty substantial clinical implications. So I'm going to now move into risk prediction using all of the information across the genome. This is an area of great excitement in the field right now. These polygenic risk scores are appealing because genetic risk is present from birth and essentially stable over time. So you can make risk assessments early in someone's life, for example. Unlike serum biomarkers, you don't have to measure them over time, like lipid levels, for example. Polygenic risk scores can also be ascertained once. So you don't have to, if somebody gets their genome sequenced and their score remains available and you don't have to resample it for their lifetime. So you can always go back to it. And then what we're learning is you can take these polygenic risk scores and combine them with other clinical factors to really enhance risk prediction. So what is it that a polygenic risk score does? Well, basically, if you think about looking across the entire genome, and in this case, we have four SNP regions, four SNPs, A, B, C, and D. Each red SNP has a small effect on a trait. Let's say it's stroke. And you basically take each individual and count the number of SNPs. So some people will have 2 copies of D, like this individual, and 1 copy of B, and you add up those SNPs and you get a score. That's essentially what the polygenic risk score does, except it does it across the entire genome. And these scores then are derived from individual association GWASs that have been performed for particular traits. So in this case, prior GWASs have been performed for stroke, so we have some way of assigning a value to each of these red SNPs. Here's a paper that was published on coronary disease just over a year ago. And what you see here is individuals were, on the right, categorized according to their genome Y, their PRS, by decile of risk for coronary disease. And then if you plot it out on the left, you can see that as your age goes up, the risk differential between those in the highest, in the blue, those in the lowest decile, and those in the highest, the red, becomes more and more spread out in terms of the cumulative incidence of coronary disease. So these polygenic risk scores are quite powerful because if they're assessed, for example, at age 60, you already have a sense of the trajectory of the individual as they age and how different that trajectory would be if somebody is in the blue range as opposed into the red range. What these authors did was then take the polygenic risk score and combine it with all of the additional risk factors listed on the left, in the left column. And you can see the odds ratios and the confidence intervals for each risk factor. And then at the bottom, you see the genome-wide polygenic risk score, which clearly has a better C-statistic than almost any of these individual risk factors other than perhaps systolic blood pressure. Then you have the combined clinical risk score, and then the combination of the combined clinical risk score with the polygenic risk score, and you're starting to get a pretty impressive statistic exceeding 0.8. So again, the lesson is you don't use the risk score in isolation, you use it in combination with other clinical risk factors. Another implication of that observation is that if we can control our clinical risk factors, we can still reduce our risk despite our having inherited a risk through the polygenic risk score. So this is a very promising and exciting area in the field, but I put this up here because I wanted to draw attention to the caveats. And these were recently published by the task force of the ICDA, which is one of these great consortia now that has brought together literally thousands of people like me studying the genetics of common diseases to come up with standards for the field, and in particular identify, in this case, some of the challenges. And I would just point you to this article. I think the main issue for us is the representation of individuals of different ancestry. So once again, these polygenic risk scores have been developed largely in individuals of European ancestry. So their performance in individuals of non-European ancestry is really questionable, and I think should be held suspect. So until we have broadly represented study samples, we won't really be able to test polygenic risk scores across a multi-ancestry population like the United States. So there's lots of work being done to improve the science to improve education around genomics and polygenic risk scores. And so I encourage you to continue to pay attention as the field evolves. So just to summarize, I've reviewed for you the way in which we think differently about rare and common variants, and how the rare variants with high large effect sizes are what are responsible for Mendelian stroke syndromes that we see running in families, generally in stroke in the young. We went over common variant discovery methods and how genome-wide association studies really have supplanted candidate gene studies going forward, and allowed us for the first time to control rigorously for what we call population stratification. I reviewed the implications for rigor, not just for genetic studies, but for human studies in general, the role of consortia, and introduce you to the ISGC, and hope some of you will go to our website and perhaps join us either virtually or in person at a future meeting. I talked about megastroke, and stay tuned for gigastroke. We talked about the importance of studying individuals from all ancestries before we can begin to really apply what we're learning in the clinic. And we talked a little bit about how we might use common variants in clinical decision making as more and more of our patients come to us with their genomes already sequenced. So thank you very much for your attention. I'd be delighted to answer questions.
Thank you so much, Jonathan. There's some online, which I'll read to you, but before we start with the online questions, does anyone in attendance have any? Come up to the microphone if you do. I have a quick one, Jonathan. Excuse my ignorance, but is the ApoE genotype different at baseline in African Americans? Like, is there overall ApoE3, E4 ratio different than in Caucasians?
Yeah, so E4 and E3 ratios differ in Africa as well, and E2 ratios do differ. The effect on the trait, though, so we do think that E4 is a risk factor for hemorrhage, probably in individuals with African ancestry. We haven't been able to study E2 yet because it's relatively rare, but this is something that has to be fixed.
Right. Because just if their baseline genotype frequency is different, ApoE2, E3, E4, that may be, and I saw they were still somewhat underrepresented in that. I had one other question just for the ICH. I was actually surprised by that slide that people with white matter disease have about this, not a really huge difference from the general population. Do you think that the general population probably also has just a lot of undiagnosed white matter disease?
That would be my explanation. And I think also when we talk about white matter disease, it's probably not one condition. So we had to reduce it to, based on the data that we sampled, to just present or absent. But there's a huge range of white matter disease. So it's a quantitative trait. So I think more accurately would be to talk about dosage of white matter disease and risk of subsequent deterioration of brain health.
Great. Okay, I'm going to read you a couple questions from the online audience. In the near future, when we genetically identify a risk for cerebrovascular disease, how do you envision this changing management? And I think you mentioned a little bit about this, but how I think probably your example of the anticoagulation will be useful.
Yeah, I think there are two things. So once the patient has a stroke, they're already declaring themselves at high risk for having another stroke. And so there are two ways in which genetics, I think, will be useful. One is it will help us to risk stratify. So, do you anticoagulate or not is one example. But also for individuals who are at super high risk of stroke, and we determine that through genomics, then those would be individuals who would be, I think, you'd want to get them into clinical trials. In other words, you really want to get high-risk individuals enrolled in clinical trials. It improves the likelihood of a successful clinical trial. So I think that's where genetics is likely to play a very big role in the near future. the long-term future, we all think that there are going to be, as we've learned with a number of the antiplatelet agents, there are genetically determined responses to different treatments.
That's an excellent point. What percentage of the heritability of stroke is currently explained by common variant SNPs?
That's a good question. Probably between 20 and 30% would be where we're at, but stay tuned for GigaStroke.
Excellent. Which brings up the next question. Is the genetic consortium moving beyond SNP arrays into exome-genome sequencing platforms to explore rare variant associations? Is there collaboration with the UK Biobank? It seems in many disease states, for example, multiple sclerosis, we are reaching the limits of common variant association studies.
Yes. So, unfortunately, we haven't reached the limits in stroke. It's just a sample size issue and heterogeneity. Multiple sclerosis has been ahead of stroke for a variety of cultural reasons, but also the disease biology, I think, is less heterogeneous. But if you think about stroke, it's really multiple conditions, so our effective sample sizes are smaller. So don't give up on common variants. However, yes, there's been a lot of work done on whole genome sequencing. Not so much has been revealed yet because, remember, those rare variants are unlikely to be discovered in even these large sample sizes because you need a lot of people carrying the variant to be sampled. We are working very closely now with UK Biobank and Biobank of Japan and all over the place. So yes, please join us virtually at the next conference.
Excellent. And we have someone on the floor, Dr. Grada, I believe.
Yep. Thanks, Louise. So, John, nice talk. What have we learned from these genetic studies about mechanisms of white matter disease? You know, what have the GWAS studies that have identified certain genes told us anything about the mechanisms of small vessel disease?
So there's a lot going on. I think the first thing is there appear to be a number of energy, so mitochondrial pathways implicated in small vessel disease. The COL4A pathways, so collagen disorders, have been implicated in lots of areas. And I think there's also inflammatory pathways that are also involved. It's a evolving field, but there are no specific treatments yet that have, I think, have come to the fore.
Okay, again from the virtual audience, is there an identified common genetic variant for stroke and sleep apnea? I'm not aware of one.
Great question. Not that I'm aware of. But it's a nice, sleep apnea is one of these, we all recognize it as an important risk factor for stroke, but the mechanisms through which sleep apnea probably lead to stroke are going to be multifactorial. And that's one where genetics might help. It might help us just disentangle some of those mechanisms. So I'd be very interested to learn when we get to, I don't follow the field, but I'm certain there are going to be genetic variants associated with sleep apnea. And as soon as they're identified, we're going to want to investigate their relationship to stroke.
Yeah, and it may be worthwhile thinking about the Sleep Smart study. I don't know if they're collecting bloods, but they are doing that study for cognitive decline. Another question, is there evidence for variability of expression of genetic risk factors, for instance, in siblings or family members?
That's a great question. So let me translate it at least into a way that I think I understand it. Are there modifiers of genetic effects? So for example, age at onset. So individuals in the family might inherit the same variant, but they have different age at onset, or some may get a more severe stroke or a less severe stroke. Yes. So what we know is that there are genetic modifiers in some of these rare stroke syndromes. And we also know that lifestyle factors affect the expression even of these rare genetic syndromes. So there's that. Even though if a sibling has a stroke, let's say, and has a Notch mutation, and their sibling has a Notch mutation, we can be fairly certain that they will develop consequences of the Notch mutation. Those consequences can very often be different.
Excellent. And the last question, because we're almost out of time, somebody asked, can you comment on potential mechanism by which E4 enhances risk of bleeding?
Well, that's been a tough one. I think Dr. McCullough is probably an expert to answer. One of the things that we're learning is that E4 is involved with vascular integrity, and it does seem like the pathological changes you see in amyloid angiopathy are related to smooth muscle degeneration. So there's got to be something involved that in the pathway of these smooth muscle cells. But lots of interesting studies are being done, and Dr. McCullough may have something to say on that.
No, it's very interesting, and it's probably tightly tied to amyloid deposition in the smooth muscle of the cerebrovascular, which is kind of a cousin to Alzheimer's disease, but it makes the vessels very friable. But that is one point I noticed in your paper looking at MRI and ApoE4. It seems like MRI is a much better risk predictor than genetics at this point,
That's
correct?
right. At this point. What genetics does in that paper is it really separates out the low and the medium risk. But the MRI really is the key determinant of the highest risk.
Right. You can think about Watchman's in those patients too, if they can't be anticoagulated. Jonathan, thank you so much for joining us. And we're going to wrap up your talk, and hopefully I'll see you in New Orleans.
Hopefully, yeah.
Come on down.
The weather has been a challenge for all of us, as you know.
Yes. All right. Take care. Thanks very much.